So I'd like to introduce Dr. Mustafa Sahin, who's going to talk about developing therapies for genetic diseases.
Thank you, Sid. I hope everyone can see my slides. These were fantastic science talks. I'm going to follow up with a much more general talk, and my task is to give you an overview of developing therapies in rare genetic diseases. First, let's start by defining what we mean by rare diseases. In the United States, this is defined as conditions affecting less than 200,000 individuals at a time. But if you take them all together, these diseases account for an estimated 300 million people all over the world. These used to be called orphan diseases because there was not a lot of interest because of their frequency from the especially commercial perspective. But during Ronald Reagan's presidency, an Orphan Drug Act was passed. And we also have a much better understanding after the genomic revolution about the biological mechanisms of these diseases. So many researchers, both in academia and industry, have been interested in these rare diseases. This is just a visual of the number of diseases increasing over the years. As you can see, we're now actually over 6,700 rare diseases identified having a genetic basis. Only about 500 of them have any kind of therapy. And importantly, roughly 3/4 of them affect the central nervous system, just like Kleefstra syndrome. This slide shows the approval of drugs for rare diseases. And as you can see, many of the approvals have been in the area of oncology or cancer. And very few, relatively few, I should say, have been in the area of brain disorders or neurology. One of the big success stories in the field, however, has been the FDA approval of a drug for spinal muscular atrophy. This was announced first around Christmas of 2016. And this drug took a disease that was essentially causing very early lethality by around 2 years of age in newborns born with this disorder due to muscle weakness. And it turned it into a chronic disorder. So children treated at the right time with this medication can now reach their motor milestones and do not die from this disorder early in life. So this was a sea change in child neurology in terms of the high impact of mechanism-based, gene-based therapies for rare disorders. I should say one of the big hurdles in this field is actual diagnosis. There are many patients out there that have neurological or developmental problems that never get diagnosed or even have the correct test done to understand whether they may have a genetic cause for this disease. So I guess about a year and a half or so ago, we put together an international consortium to review the literature in the field about the diagnostic yield of using some of the advanced tests, such as the exome sequencing for neurodevelopmental disorders. And Dr. Sid took the lead on this paper that's been published. And this paper really makes the argument that individuals with neurodevelopmental disorders should have access to exome sequencing as early as possible. And with this kind of approach, more and more individuals will be identified early in life with the genetic cause of the disorder. This is really important, especially for rare disorders, where you want to have a larger cohort and understand the whole spectrum of the disease and its natural history. This is a graph that shows the publications, papers, scientific papers in the field of rare genetic disorders. As you can see, this is a very huge increase, 100-fold increase over this time frame from 1980s to our time right now. Unfortunately, we have not seen the same increase in the number of drug approvals. And that really shows the unmet need in between these two lines you can see here. The approach we take at Boston Children's and many of our sister centers is to start with genetically defined neurodevelopmental disorders. And we have subspecialty clinics for many of these disorders, and Cleve's clinic will be joining those subspecialty clinics. We do phenotypic characterization of the patients very carefully. As Dr. Sid mentioned, it's really important to understand the symptoms and diagnose them correctly. But we also oftentimes try to use more quantitative markers of the disease. In some cases, this can be EEG based. In other cases, it can be imaging based, such as MRI. In other cases, it can be based on a blood biomarker. And then we, as Dr. Kastri and Dr. Buttermore mentioned, we try to use cell-based models of these diseases. In the past, we used to use mouse and rat neurons. More and more, we are using human neurons, as you saw earlier. And then almost always, we try to go to an animal model, usually a rodent, to test the safety and efficacy of the therapies we've identified, and then go to clinical trials. And in many, many cases, we have to go through this cycle more than once to find safe and effective medications or interventions. I'm going to talk in this very broad talk about different types of drugs that can be potentially developed for rare diseases. This is from a recent review. It talks about the possibility of using small molecule drugs, protein replacement antibodies, something called oligonucleotide therapies, or ASOs you may have heard of, and then cell and gene therapy. I'm mostly going to talk about small molecules, oligonucleotide therapy, and gene therapy. I'm not going to talk much about protein replacements or antibodies for the purposes of this conference. If there are any questions on those, I can address them later. So small molecules are the sort of prototypical drug that you buy from your drugstore these days. These are the best established therapeutic modality. These are small molecules. They are low cost. They're relatively easy to synthesize. You can adjust the dosing relatively easily, up or down. You can give it orally. You can give it intravenously. There are multiple routes of administration. And they are quite easy to absorb, and they go into the different organ systems in relatively easy to predict ways. So there's a lot of chemistry, medicinal chemistry known about this group of drugs, and they have been in use for decades now. Now, to develop new small molecules, oftentimes what we have to do is to do a screen, and we have to use a large library of chemical entities, basically new compounds that are synthesized or obtained from nature and modified. And in most cases, this library of compounds is used to interrogate an assay. This can be a test tube assay, whether to see if those compounds bind to a certain protein, or it can be a cell culture assay, similar to what Drs. Buttermore and Kasi were showing earlier, to see if they make a change in the behavior or shape of the cells that you're studying. As you heard earlier, the stem cell-derived neurons, as well as this gene editing technology called CRISPR-Cas9, is really changing the landscape. In the past, especially for brain disorders, we used to use cell lines that probably had nothing to do with the disease itself in terms of not being nervous system related. They were used for high throughput screens, but more and more we're actually able to make the type of cell that is most relevant to the disorder and test this chemical library on that group of cells in culture. But you can also, as you heard earlier about fruit fly models, there are other models such as yeast that can be used to develop assays, and they are still in wide use right now. The pluses of developing small molecules is a price. They're usually cheaper when they go into the market. The scale is easier, and the chemists and the pharmaceutical companies are actually quite expert for developing it for certain class of targets, especially enzymes such as kinases, this group of molecules, that are especially important in cancer, and also for cell surface proteins. But there are a number of other targets in the cell that may not be easily targetable with the small molecules. And then the minuses of this approach is that it can have off-target, basically adverse events related to the use of these drugs. Even though they may affect the target, they may also have effects on organs that are not necessarily the target of choice there. You really need good assays to be able to develop effective drugs, and you need large chemical libraries. For many of these reasons, large pharmaceutical companies have become really the best approaches to develop these drugs. Academic centers like ours can develop potentially targets and tool compounds and assays, but we often have to collaborate with our colleagues in the industry to have drugs that will actually come to the market. There's another approach in the small molecule realm that is quite important, especially for rare diseases, and that is repurposing drugs. These are drugs that have been approved and developed for a certain indication, but then you take that drug and use it in a different disease and a different condition. The really big advantage of this is that most of these drugs have well-established safety and dosing profiles. You know how much to give to the patient, and you know what side effects, if any, they're going to have. So that really accelerates the development time and the risks of doing the trials. So I'm just going to briefly mention the time it takes to develop a brand new drug. I'm not going to go into all these different steps, but basically you can start from very basic science studies, and you move more and more to finding one compound that you think is going to be successful in clinical trials. Then you go into the clinical phase that can take up to 5 or 6 years, and then that leads to approval by the registration agencies. That can take 10 to 17 years, and the chance of success in this particular review was estimated to be less than 10%. If you take a repurposing approach, you take a drug that's already been used and in clinic for a different indication and try it in a different disorder, that can significantly reduce the time it takes to get approval. So it reduces the screening steps that you have here. It reduces the actual licensing aspects, etc., and you can initially start from the clinical stage relatively quickly, and the chance of success is potentially higher with relatively reduced risk. As you can see, the time frame estimated here is 3 to 12 years. So this I hope gives you a sense of the time frame it may take to develop a brand new drug or taking a repurposing approach. This is just comparing the two approaches, as I just mentioned earlier. I just want to give you a very quick example from a disease that we study quite a bit called tuberous sclerosis. You, Dr. Buttermore, already. This is a disease that causes benign tumors called subapentymal astrocytomas, epilepsy, and autism in patients. These genes that are known to cause TSC are due to loss of one copy of the TSC1 or TSC2 genes. What happens when those genes are lost is a kinase called mTOR, which stands for mechanistic target of rapamycin, becomes hyperactive, it becomes too active, and that's the cause of the disease. It turns out in nature, we've been given a very specific inhibitor of this kinase, of this enzyme, and that was found by serendipity in the soil in this Easter Island in the Pacific Ocean. Soil samples from this island led to identification of a compound called rapamycin. Here's the chemical structure of this compound. This compound was initially thought to be an antifungal. It killed fungus, but it turns out that it's a better immunosuppressant. And so, over 20 years ago, rapamycin got FDA approval as an immunosuppressant for organ transplants, like kidney or heart transplants, and it's still currently used for those indications. But then more recently, it's shown to be effective for improving the stent staying open in heart disease. So there are now stents that come with a compound similar to rapamycin called everolimus to the stent. And then a certain formulation of rapamycin-like compounds called Afinitor got approval for treatment of renal cell carcinoma. All of these compounds basically inhibit the mTOR pathway. So the idea was, can we use it for tuberous sclerosis patients? And the first studies were done in these benign tumors, astrocytomas, with just 28 patients. The study was done, and in all of the patients, the tumor shrunk or stayed the same, it did not grow. So that led to the FDA approval of this compound around 2010. More recently, it required a study of about 300 patients to show that we could improve the epilepsy in those patients with tuberous sclerosis and highly intractable epilepsy. And that was approved about 3 years ago. Unfortunately, we've done some studies looking at behavioral and cognitive endpoints in patients with tuberous sclerosis and autism, and we have not been as successful in showing the efficacy of that group of compounds. So even a compound may work, a small molecule may work for certain indications. It might be harder for other indications to show that they actually are effective as well. Now, I want to briefly talk about oligonucleotides. You may have heard of these recently. They work at the level of RNA. As you know, we go from our DNA that gets transcribed into RNA, and then RNA leads to the making of the protein. There's a group of oligonucleotides called antisense oligonucleotides. They bind to complementary RNA. They often lead to degradation of that RNA. So if you have a RNA that's toxic or something that's overly expressed in a cell, you can break them down using these ASOs. It can also result in exon skipping or alternative splicing. So there are some variants that cause disease. In those variants, if you skip the abnormal exon, you can have a gene expression that's much more functional. And then alternative splicing is another way to either increase or decrease the amount of functional protein. These can be very specific. And the advantage of this approach is that unlike the small molecules, which work well for a certain type of proteins in the cell, you could theoretically use this approach for any gene of interest. The certain variant in the gene has to make sense in terms of this approach. So I don't have time to get into the details of that, but this approach has been now used. And the approval that I mentioned in spinal muscular atrophy was actually one of these oligonucleotide approaches. So it actually, this drug called nusinersen is an ASO-like drug that works on improving the expression of the protein that's missing in these cells. The plus is that they can target any gene, and the other plus is that it can be reversible. So even if you had side effects, these will eventually wear off, and you'll be able to come off the drug. Minus is that you may need a different drug for different patients with the same disease because of this particular sequence specificity that I mentioned earlier. Another downside potentially is that they do not usually cross the blood-brain barrier. And then the approach usually requires repetitive dosing. So for brain disorders, you may need a spinal tap every few months. And the final thing I want to talk about is the gene therapy. For this, we have to get the gene that's, in this case, often missing into the organ that's affected. In that case, probably the brain cells, the nerve cells. For neurons, an approach that seems to be particularly effective is using adeno-associated virus. This virus has certain advantages in terms of getting it to neurons. It's still not an optimal virus. It probably needs some improving. One downside of this particular virus approach is that it has a certain limitation in terms of how much DNA you can put into it. So the gene size does matter. FDA has actually approved this for another drug that was developed for spinal muscular atrophy. they managed to put the SMN gene into this type of vector. And just one dose of this virus injected to a baby with SMA seems to have long-lasting effect without requiring repetitive dosing. And those children also do extremely well, just like the oligonucleotide approach I mentioned earlier. I want to highlight the paper that the Kleefstra published about the identification of EHMT1 as the cause of Kleefstra syndrome. In that paper, obviously, she showed that heterozygous, or losing one copy of the EHMT1 gene, is sufficient to get the disease. And in that case, the variants that she's identified seem to cause loss of function. So if there's a way to increase the amount of functional EHMT1, this is the approach that we want to go after. So this is potentially a way of thinking about gene therapy for Kleefstra syndrome and related disorders. The approach has a significant advantage in the sense that it's a one-time therapy, and you get long-term expression of that gene, and you get the right gene, correct gene, without the mutation expressed. So in some ways, this is the cure that we are all hoping for. Minus is that you have to get into a large number of cells, and that's a significant limitation with the vectors that we're using right now. In some diseases, having too much of the gene can be toxic. It can have side effects, so one has to be careful. It can be difficult to manufacture. It's not as easy as manufacturing small molecules or oligodendrocytes. And there's another aspect that I won't dwell on right now, but you can have an immune response to the expression of either the protein you're expressing or the viral vector that you're using, which limits some of the uses in certain patients. So I went through a lot of different approaches. In all cases, we almost always start with the identification of the gene, the preclinical models. We need natural history studies, observational studies, to be able to move into the clinical trials. The early phase clinical trials, phase 1 and phase 2, are really to test the safety of the drugs, especially if they are new drugs that are not being repurposed. This is a really important phase. And then we finally showed the efficacy in larger phase 3 trials. And this can seem like a very long time, as meant to show with this photo. I just want to highlight that there are significant hurdles in this process. Fragile X syndrome, which is a common cause of intellectual disability, has been a disease that's been studied by many investigators, and they identified a group of compounds, in this case, a type of glutamate receptor antagonist, to be very effective in mouse models, also effective in fruit fly models, in cell culture and in vitro models. However, when they went to the clinical trials, they were unable to show efficacy of these drugs. So the investigators here conclude, and I'm just copying here, preclinical studies or results suggest that future clinical trials might profitably explore initiating treatment in a younger population with a longer treatment duration, a longer placebo run-in, and identifying new markers to better assess behavioral and cognitive benefits. So the bottom line is that sometimes, even though things look very good in the early stages, to show that they work well in final stages may make some improvements in clinical trial design. So I'm going to briefly summarize here what we've learned from other rare disorders. The trials aimed at improving cognitive and behavioral symptoms are usually more difficult than trials with objective outcomes, such as survival or tumor size. Biomarkers, imaging or electrophysiology, or blood biomarkers are very important, and improved outcome measures, things you can quantify and measure and that change over a relatively short period of time, are really important to be able to detect meaningful change in these clinical trials. Having an in-depth longitudinal natural history study can accelerate clinical trials dramatically. So I'm very excited about the natural history studies ongoing in Kleefstra syndrome. The other consideration is that the timing of the intervention for the individual enrolled might be critical. for certain diseases. The distribution of the therapy, which brain region the therapy goes to, what cell type it can get into, might be also crucial. And then the dosage of gene expression per cell might be essential for certain disorders. And we can talk about how these affect EHMT1 related disorders. Oh, so I'm going to stop here. I want to thank our patients and their families, which have motivated and guided our research. A number of folks from the Translational Neuroscience Center are shown up here. Liz Buttermore and C.D. Chen, who run our Human Neuron Core, and various collaborators, including Dr. Clifstra, and our funding sources. Thank you so much.
Excellent. I think we probably have time for one question, and I think just one of the themes that seems to be coming up is differences between those who have chromosomal deletions with many genes affected potentially versus those with single gene mutations. So I was wondering if you could talk a little bit about considerations for developing therapies for those two different types of molecular populations.
Yeah, in general, and I'm going to again talk in general rather than Clifstra specific at this point, but diseases in which we can identify one gene and can change the expression or functionality of that gene are going to be the easiest disorders to go after first. We don't have a lot of mechanisms currently to address the disorders in which a large number of genes are misexpressed or misfunctional. And if that's the case, those individuals may not respond to a treatment that affects only one gene's function. They may still be, participants in a trial, but we may not be able to expect the same amount of efficacy. So there's a number of copy number variations, deletions, and duplications being identified that affect the central nervous system. And many of them, we don't know exactly what gene is the cause. In the case of Clifstra, I think we have a very good idea. But the severity, as we heard earlier, may differ depending on the size of the deletion. So we may have to stratify in future clinical trials which patients are eligible for which therapy.
Excellent. Well, thank you so much, Dr. Sahin.